And just recently, we have started working with Saurabh Mahandru, who's made this rather remarkable observation that the virus seems to be persisting in the gut perhaps longer than we had previously appreciated. Some of our clinical work on the clinical samples has been done in collaboration with New York Blood Center, particularly.
So my next one, I'm not necessarily going to fight this battle with you, but I want to pass on that 2 of our previous speakers have given some data that's highly suggestive that persistent infectious virus lasts. So most recently we had Paul Binash, and if people want to look it up, it's the November 11th talk. He had 2, I hope I'm not going to, misrepresent what he said, but my understanding of his data set showed 2 really salient things. One, they were seeing in fully a third of their patients, they were histologically detecting spike protein in gut after 6 months. Which, I mean, it could be debris, but that's a long time.
That's a long time, yeah.
For debris to last. But then what I thought was the most compelling evidence for a live reservoir of virus is they sampled convalescent plasma from patients in a longitudinal fashion. And then they used that to try to neutralize specific strains of the virus, mostly wild type, and then a combination of 2 antigenic escape mutations. And they found that initially the patient's convalescent plasma did not neutralize the antigenic escape mutations, but then after 4 to 6 weeks, it did, suggesting that, in my mind, the only way that makes sense is if there is a pool of virus.
In terms of potency and breadth. So something is going on for a long time. And you can see here from this
what's going on. There's always this sort of uncertainty whether you find something by PCR, does that really reflect viral replication? I mean, even if you see an immunofluorescence picture of a cell with viral antigen in, is that really an infected cell or a cell that's just eaten some protein? My suspicion is it's real virus replication.
In the end, this is probably the most surprising finding from the study, which was that memory B cells, people had more memory B cells at 6 months post-infection than they did at 1 month post-infection. And that was true for spike-specific memory B cells, RBD-specific memory B cells, as well as nucleocapsid-specific memory B cells as an independent antigen. And it was also true for the longitudinal data from per individual. And it was also true if we specifically looked at the IgG-specific, spike-specific memory B cells, that there were more at 6 months than 1 month. This is consistent with an earlier finding by Marion Pepper's lab, where she observed more at 90 days than 30 days. And it's also consistent with a paper just published by Michel Nussensweig's lab, looking at specifically a 6 month time point compared to a 1 month time point, and they saw the same type of increase.
I guess the thought from that is that how would these viral fragments remain around for that long? Right?
If they're not replicating.
We chew up everything, don't we? They would have to be very highly resistant to degradation or protected by some other mechanism. And so otherwise, it's hard to imagine. I'm not saying there is replication recombinant virus, Brianne. I'm just questioning, how could we retain those fragments around for 6 months?